Welcome to Biochemistry. Today, we are going to explore the fascinating interconnection of key metabolites. If you have ever wondered how your body knows whether to burn fat, store sugar, or use protein for energy, you are in the exact right place. We will build this understanding from the ground up. Let us dive in. To begin our journey, imagine standing in the middle of a massive, bustling international airport. Passengers are arriving from all over the world, and they are all trying to reach completely different final destinations. In your body, your liver acts exactly like this grand central airport. The passengers are the carbon molecules from the food you eat. Instead of airport terminals, your liver uses three major biochemical intersections to route these molecules efficiently. Understanding how these specific hubs operate is the absolute core secret to how your body manages energy, stores fat, and keeps you alive. Now that we understand the airport analogy, let us look at our first major terminal, glucose 6-phosphate. This molecule is the primary hub for carbohydrate metabolism. Think of it as the main gateway for any sugar entering your system. Once glucose becomes glucose 6-phosphate, it faces a choice. It can be stored away as glycogen for later use, broken down immediately for quick energy, or even used as a building block to make structural sugars. It is the ultimate decision point for carbohydrates. Moving deeper into the metabolic pathways, we arrive at our second major hub, pyruvate. Pyruvate is known as the versatile middleman of the cell. It serves as the critical crossroads where the metabolism of carbohydrates directly intersects with the metabolism of amino acids derived from proteins. This means that if your body is under stress and needs to, it can take the building blocks of your muscles and convert them into this central molecule, effectively linking two entirely different types of macronutrients together to keep you functioning. Finally, we reach the ultimate convergence point of human metabolism, acetyl-CoA. No matter what you eat, whether it is a bowl of pasta, a piece of steak, or an avocado, the carbohydrates, proteins, and fats are all eventually funneled down into acetyl-CoA. From here, the cell makes a final crucial decision. It will either burn this acetyl-CoA in the mitochondria to produce massive amounts of cellular energy, or it will use it to synthesize new fatty acids for long-term storage. Why is this interconnected system so incredibly vital for our survival? Imagine if our bodies had completely isolated pathways for sugars, fats, and proteins. If you ate only protein, your brain, which relies on glucose, would rapidly starve. But because of these hubs, your liver can take amino acids, convert them into pyruvate, and run the machinery in reverse to manufacture brand new glucose. Conversely, eating excess carbohydrates funnels sugar down to acetyl-CoA to build body fat. This metabolic flexibility allows humans to survive diverse diets and prolonged fasting. While these hubs form a conceptual map, we can actually mathematically quantify their energy value. The most important calculation in metabolic biochemistry is determining how much usable energy, in the form of ATP, is extracted when a molecule passes through the acetyl-CoA hub. We use a specific stoichiometric formula for this. Y represents the total ATP yield. N is the number of NADH molecules, which carry highly energetic electrons. F is the number of FADH2 molecules. Finally, A represents any ATP or GTP produced directly. Notice that each NADH yields 2.5 ATP, making it a massive driver of energy. Let us see this formula in action by calculating the total ATP yield when one molecule of pyruvate is fully oxidized. First, we map the biochemical steps. Converting pyruvate to acetyl-CoA gives us exactly one NADH. Then, running that acetyl-CoA through the citric acid cycle yields 3 more NADH, 1 FADH2, and 1 GTP. Adding our NADH together, we get an N value of 4. We plug these into our energy yield equation, multiplying 4 by 2.5 and adding the rest. We find a massive yield of 12.5 ATP. This high energy output is exactly why our bodies rely on this pathway during aerobic exercise. Now that we have calculated the energy, let us shift our focus to the physical matter itself. How do the actual carbon atoms flow between these hubs? When a cell takes in a molecule of glucose, it physically breaks it apart, routing the pieces through pyruvate and acetyl-CoA. But matter cannot simply disappear. By tracking the total number of carbon atoms at each stage, we can account for every single piece of the original sugar molecule, including the atoms that are lost as carbon dioxide gas during the process. To track these physical atoms, we use a simple carbon mass balance equation. In this formula, C represents the initial number of carbon atoms in our starting molecule, which for glucose is exactly 6. The variable K stands for the number of acetyl-CoA molecules formed at the end of the pathway. Because acetyl-CoA is a 2-carbon molecule, we multiply K by 2. Finally, the variable M represents the number of carbon dioxide molecules released into the bloodstream. If the number of acetyl-CoA molecules increases, the remaining carbons available to form carbon dioxide must decrease to keep the equation balanced. Let us apply this mass balance equation to a real scenario. We know that one 6-carbon glucose molecule splits into 2 pyruvates, which then convert into 2 acetyl-CoA molecules. Therefore, our K value is 2. We substitute our known values into the equation, setting 6 equal to 2 times 2 plus 1m. The 2 acetyl-CoA molecules account for exactly 4 of the original carbon atoms. This leaves a mathematical gap. Solving for m, we find that exactly 2 molecules of carbon dioxide are produced. This beautifully illustrates that when you burn sugar, you literally breathe out the broken down carbon atoms into the air. Let us bring all of this together visually. Here, you are seeing the complete flowchart of our metabolic hubs. At the top, glucose enters and immediately hits our first hub, glucose 6-phosphate, which can branch off to the right into glycogen storage. Moving down, we hit pyruvate, which you can see intersecting horizontally with amino acids. Finally, the pathway funnels down into acetyl-CoA. From acetyl-CoA, the arrows diverge again, pointing right toward fatty acids for storage, or continuing downward into the citric acid cycle to generate the massive ATP yield we calculated earlier. This is the grand central station in action. We have covered a lot of ground today, so let us review the most important ideas. First, always keep your eyes on glucose 6-phosphate, pyruvate, and acetyl-CoA. They are the grand central stations seamlessly routing carbohydrates, fats, and proteins. Second, these interconnected hubs are what give the human body its incredible metabolic flexibility. Third, we learned how to mathematically quantify the energy yield from these pathways. And finally, how to track the physical carbon atoms that we eventually exhale. You now understand the fundamental logic of human metabolism. Great job!